In this video, I just want to introduce some of the basic anatomy of the cell. Now, we all start from a single cell, just one fertilized egg. That cell divides and divides and divides and divides and divides and divides, and as more and more cells accumulate, they start to take on distinct characteristics. This is a process called differentiation. Just basically cells becoming different from one another. Eventually, we end up with cells that are very different. They look different, they have different functions, they can be very highly specialized. For example, you have the skeletal muscle fibers. That's what you call skeletal muscle cells. They're called fibers because they are so long and stringy. And they are long and stringy so that they can contract over a significant distance and move your body. Neurons have long extensions called processes that allow them to send signals from your brain or spinal cord all the way through your body. You have red blood cells, which lose their nucleus and are a little bit smaller so that they can fit through the smallest of blood vessels. White blood cells that are bigger, and they have their nucleus because they need to make lots of proteins to help protect your body from infection. Now, we're not going to worry too much about the specialized cells right now. We'll get into those when we get into specific organ systems. In this video, we're going to be talking about just kind of a generalized cell. We are going to focus on a generalized animal cell. Plant cells have some differences, but since this is a human anatomy and physiology class, we're not going to talk about plant cells. This is just about the most generalized and simplified version of a cell you'll find. We have the cell itself. Actually, technically, this is the plasma membrane. The plasma membrane is the border of a cell, what separates the inside of the cell from its environment. And that way you can kind of sort of think of the plasma membrane as like the skin of your body. So you've got the plasma membrane. You have the nucleus, the control center inside the cell. This is where the majority of the DNA in the cell is. And then in between the two, in between the nucleus and the plasma membrane, you have what's called the cytoplasm, and that's the semi-fluid interior full of organelles. I've arranged this video according to these 3 cell compartments. So we'll talk in a fair amount of detail about the plasma membrane first. Then we'll talk briefly about the nucleus, and we'll talk about the cytoplasm and the different organelles and structures within the cytoplasm. First off, I want to talk about the general structure of the cell membranes. And when I refer to the cell membranes collectively, I mean both the plasma membrane, which remember is the external membrane that wraps around the cell, and internal membranes. So some of the organelles have their own membranes, or are their own membranes, like the Endoplasmic Reticulum, the Golgi apparatus, the nuclear membrane. They all have the same basic structure, although the details vary. And that basic structure is the phospholipid bilayer. So if you remember the kind of hairpin structure of a phospholipid, with the hydrophilic head and the 2 hydrophobic fatty acid tails, these phospholipids line up tail to tail, right? So, and they line up forming this, basically this sheet that wraps around the cell of these tail to tail, these phospholipids stacked tail to tail. So you have this hydrophobic center where the tails are, and you have a hydrophilic layer where the heads are, both on the outside of the cell facing into the external cell, external environment, and on the inside of the cell facing into the internal environment of the cell. So the phospholipid bilayer is the basic structure of a cell membrane, but it's not the end of the story. You also have molecules inserted into this layer, this bilayer of phospholipids. You have cholesterol molecules. If you remember, cholesterol has that 4-ring shape. You have cholesterol molecules inserted into the membrane. They help stabilize the membrane. You also have proteins embedded in the membrane as well. Proteins can either be embedded in the phospholipid bilayer so that they're actually stretching through the membrane. These are called integral proteins. But you can also have proteins that are just kind of sitting up against the bilayer. They aren't actually within the phospholipid bilayer. And when they're just sitting up against, kind of on the periphery of the membrane, they're called peripheral proteins. Proteins at the membrane, either peripheral or integral proteins, have a huge variety of functions, particularly if you're talking about the plasma membrane. This slide summarizes what some of those functions are, and I want to talk just a little bit more about each of them. The first function is just attachment. Cells need to be attached to their extracellular environment. You might think of the extracellular environment as just being this kind of fluid, but it's actually more sort of gelatinous. And it's filled with proteins, primarily collagen fibers. We'll talk about this more when we talk about the connective tissue in particular. And cells that stay in one place need to be anchored to this extracellular matrix of collagen fibers and other molecules. So integral proteins in the plasma membrane do this anchoring. They connect to the collagen fibers and hold the cell in place. They also connect to the cytoskeleton, and that's what these little orange ropes are. They're kind of the internal framework of the cell. And so some proteins kind of act as a bridge. They hold the plasma membrane against the extracellular matrix. They connect the plasma membrane to the cytoskeleton as well, so that the whole cell is held in place and doesn't rip apart. Some membrane proteins function to attach cells to other cells. Points at which cells come together are called cell junctions. And there are a few different types of cell junctions. I've just illustrated here a couple that are important. We'll talk a little bit more about this when we get into discussing the tissues and we discuss epithelium. Right here you're seeing an electron micrograph, and you can actually see, you're looking at the surface of a cell, and you can see the proteins in the membrane of that cell. And these proteins would connect to a neighboring cell. Connect to proteins in the surface of a neighboring cell. Over here, you are looking at different types of proteins forming a different type of cell junction called a desmosome. But you see that shadow right there? Let me illustrate it for you. You've got one cell there, and another cell there. And you have proteins from one cell linking up to proteins in another cell, sealing those two cells together. Proteins at the plasma membrane can also act as enzymes. Remember that enzymes are proteins that catalyze chemical reactions, that speed them up. Plasma membrane proteins are also involved in cell-cell recognition, particularly glycoproteins. And remember that protein, that term just means that you have a protein with a little carbohydrate tag attached to it. And these glycoproteins, they often serve as cellular ID tags. They allow specific types of cells to recognize each other and interact. One absolutely critical function of plasma membrane proteins is to function as signal receptors. The plasma membrane is where a cell comes into contact with its environment. And membrane receptors are how a cell detects changes in that cell's environment. Your body's control systems, the nervous system and the endocrine system, both depend heavily on membrane receptors. Membrane receptors are so very critical right? First thing I want to say about membrane receptors is that shape is really critical for a membrane receptor. There is kind of a lock and key relationship between a signaling molecule and the receptor for that molecule. You see how the signaling molecule fits right into a site on the receptor? That's a very specific fit. So this receptor is only going to bind one type of signaling molecule. Now, the cool thing about having these membrane receptors is that the signaling molecule doesn't have to actually get into the cell. The signaling molecule stays on the outside. But what happens is that when the signaling molecule binds the receptor shifts. It just changes shape a little bit, and changes shape in such a way that it causes a cascade of reactions to start out on the inside of the cell. So the signaling molecule, through binding here at the receptor, causes the receptor to shift, and that triggers a set of reactions within the cell that can cause a change in the cell. It's a little bit like, if you remember when we talked about enzymes, we talked about how a non-competitive inhibitor can bind to an enzyme and cause the enzyme to change shape so that the substrate doesn't bind anymore. Signaling molecule can cause a receptor to change shape. Only in this case, that change in shape stimulates a set of responses within the cell. The last function of membrane proteins is to allow for transport of substances across that membrane. Like I said, the plasma membrane is where a cell comes into contact with its environment. So if the cell wants to bring something in from its environment or expel waste out to the environment, it has to cross the membrane. And many substances, the membrane only allows certain substances to pass through that phospholipid bilayer. So other things need transport proteins to help. Now, when we discuss membrane transport, we'll go into a lot more detail about the different types of membrane transport and a little bit about the different types of transport proteins. Just for now, understand that some things can pass straight through the phospholipid bilayer, but most substances need proteins embedded in the plasma membrane to help. The last thing I want to say about the anatomy of the plasma membrane is pointing out that some cells in the body have projections, extensions of the plasma membrane that stick out from the main body of the cell. And there are three types of cell extensions. We have the flagella, cilia, and microvilli. Now, flagella, there's only one type of human cell that actually has a flagellum, and that's the sperm cell. So really only about approximately half of the human population actually has flagella, but it's kind of important. And a flagellum is a single long, like a tail on the sperm cell. I want to show you a brief video of some mouse sperm cells. The human sperm cell does not have this hooked head like the mouse sperm cell does, but you can see the flagella and see how the flagella beats to propel the sperm cell forward. So a flagellum is a single long extension on a sperm cell. When a cell has either cilia or microvilli, they have multiple, just coating a single surface of the cell. Just one surface, though. So if you have a cell, the cilia or the microvilli are just going to be found on one surface. And we'll talk about why that is when we talk about the epithelium. Now, cilia and microvilli look very similar, but they are not the same. They have very different functions. Cilia actually move. They beat back and forth, setting up a current that sweeps mucus across the cell surface. In your respiratory tract, it's cilia on your airway cells that sweep debris up and away from your lungs. I want to show you that. So here's your airway cells, and here's the cilia. Now, watch what happens when I hit play. Did you see them moving back and forth? In contrast, microvilli are stiff. They don't move. They're just there to increase the surface area of the cell to better absorb substances, like in your intestine, in your small intestine, to better absorb nutrients out of your food. Here I'm just showing you a visual comparison between cilia and microvilli. They both form this kind of fuzzy appearance on the surface of the cells that have them. So there's the cilia, there's the microvilli. But cilia move, so they look a little bit less even. Notice how it's not quite a perfect little border. There are some kind of gaps and wavy-like appearances. The microvilli, however, they're stiff, and they stand up stiff like the bristles of a brush, so they have a very, very regular appearance. And in fact, this is sometimes called the brush border. Okay, now I'm going to talk about the nucleus. And the nucleus is a highly specialized organelle. It's where the vast majority of the DNA in a cell is located. It's the DNA in your nucleus that comes half from your mom and half from your dad. But the nucleus isn't just a passive house for DNA. The nucleus also actively participates in DNA regulation. It is surrounded by a membrane, the nuclear membrane, and the nuclear membrane is actually very important in organizing the DNA and making sure that the DNA is used correctly. The nucleus is also the location where DNA is transcribed into RNA, the first step in making proteins. The vast majority of cells in your body have one single nucleus, like the cell on the previous slide. However, there are some exceptions. Very few cells have multiple nuclei, and the term for that is just pretty easy to remember. It's multinucleate. Skeletal muscle is one example of a multinucleate cell. So if you look here, I'm going to outline a single skeletal muscle cell there. And just in this tiny little slice of the skeletal muscle alone, you can see 1, 2, 3, 4, 5, 6 nuclei. And skeletal muscle fibers can have hundreds of nuclei. There is also a type of cell in your body that doesn't have a nucleus, and that's your red blood cells. Red blood cells, the term for that is a nucleate. It's only mature red blood cells that don't have a nucleus. They actually do have a nucleus while they're developing, but they kick it out as they age. So this serves a couple of different purposes. It gives them more room within the cell to carry hemoglobin, the molecule that actually carries oxygen, and it also allows the red blood cells to be more flexible so that they can move through the tiniest little blood vessels without getting stuck. While we're on the subject of the nucleus, I also want to talk a bit about the structure of the DNA in the nucleus. You know by now that DNA has this double helix structure, but the DNA has to be really carefully packaged up in order to fit inside the nucleus. You have a ton of DNA inside your nuclei. So you take this basic double helix, but then the double helix is wound around packaging proteins called histones. This packaging, this wrapping of DNA around the histones, gives DNA kind of a beads on a string appearance if you look at it under the electron microscope. And the term chromatin, like they're using here, that's basically DNA, so the molecule DNA, plus its packaging proteins. But we're not quite done wrapping that DNA up yet. The DNA wrapped around its histones is then wound up on itself kind of in a spiral. And this is how DNA is packaged basically in non-dividing cells. It's at this level, wrapped around the histones and then twisted into a spiral. when a cell is getting ready to divide, it becomes really, really important that the chromatin become as tightly wrapped as possible, because you don't want chromosomes breaking apart in the middle of cell division. That would be very bad, and very bad things do happen when that occurs. So if a cell is getting ready to divide, the chromatin becomes even more tightly wrapped up, and it condenses to the point where you can actually see it visibly under the microscope. So this level of wrapping and condensation, that's what chromatin looks like in cells that are preparing to divide. Okay, so we've talked about the outer layer of the cell. We've talked about the control center of the cell, where the DNA is. Now let's talk about the stuff in between, the cytoplasm. In the cytoplasm, you find a lot of different things. You find cytosol. Cytosol is the fluid portion of the cytoplasm. It's kind of the substance that everything else sits in. You also find lots of organelles. Organelles, think of them as the little organs of the cell. They are the metabolic machinery of the cell. They're like little structures within the cell that carry out particular functions. You also sometimes find inclusions. And inclusions are just inert storage structures. They're not present in all cells, but if you have a cell that needs to store a substance, like, say, glycogen in your muscle cells, then that substance is stored in an inclusion. The last thing that you find in the cytoplasm is the cytoskeleton, and the cytoskeleton forms the framework of the cell. It's a network of protein fibers that give shape to the cell, and they also serve as highways to move material through the cell. This is just a quick picture illustrating some of the important cytoplasmic structures that you will see. If you take a look in your textbook, there's a spotlight figure, 3.1, that goes into a lot of detail about the cytoplasmic structures and their functions. Mitochondria are the power plants of the cell. And mitochondria, yes, it's plural. The singular form is mitochondrion. It's usually used in the plural because you have many mitochondria in a single cell. And some cells, like cells that need a lot of ATP, like your muscle cells, have hundreds to thousands of mitochondria in a single cell. And so these guys are the power plants, and when you think power in the cellular realm, you should think ATP. So mitochondria are your ATP producers. This is where fatty acids and glucose can be broken down to form ATP. And mitochondria also contain their own DNA. They're the reason why I say that the nucleus contains the majority of the DNA of the cell. But mitochondria have their own DNA and their own RNA and ribosomes, and it's actually thought, it's one of the lines of evidence for mitochondria having once been free-living bacteria, that some eukaryotic ancestral cell way back in evolutionary history took in, and they formed a symbiotic relationship. You also have the endoplasmic reticulum. The endoplasmic reticulum is a network of membranes, and it actually forms about half the cell's membranes. There are two types of endoplasmic reticulum, or ER. You have the rough ER, and you have the smooth ER. And the rough ER is rough because it's studded with ribosomes. It's the ribosomes that make the rough ER rough. And ribosomes are absolutely critical for making proteins. So one of the major functions of the rough ER is making lots and lots of proteins. You do have ribosomes that aren't attached to the rough ER, but a lot of them are. In the smooth ER, it's smooth because there's no ribosomes. So protein synthesis is not a major function, but the smooth ER does do a number of different things. It's involved in drug detoxification, making some hormones like the steroid hormones, making lipids. So there are cells in your body that are specialized for these functions that contain enormous amounts of smooth ER, like cells in the liver, which does a lot of drug detoxification, muscle cells, hormone-producing cells. These all have lots of smooth ER. Next we have the Golgi apparatus, also known as the Golgi body or the Golgi complex. And the Golgi apparatus is a stack of membrane sacs, like someone stacked them up and then flattened them down. And in here you see a lot of processing and sorting of proteins. This figure shows the relationship between the endoplasmic reticulum and the Golgi apparatus. It's comparable to figure 3.7 in your book, another big splashy spotlight figure with a lot of good information in it. So notice that the Golgi apparatus is located between the endoplasmic reticulum and the plasma membrane. There's this progression. Proteins that are made in the endoplasmic reticulum pass through and are sorted in the Golgi apparatus. And from the Golgi apparatus, they oftentimes go to the plasma membrane to either become a part of the membrane or to be secreted. Proteins also may leave the Golgi apparatus and become located in vesicles in the cytoplasm, particularly lysosomes. When proteins are formed in the ER, they leave the ER in these little transport vesicles. And a vesicle is basically just a droplet of membrane filled with substances, filled with proteins. So the vesicle carries these proteins from the rough ER to the cis face of the Golgi apparatus. And it's called the cis face because it's closest to the rough endoplasmic reticulum. It's the side that faces the rough endoplasmic reticulum. Proteins pass through the Golgi apparatus to the trans face of the Golgi apparatus, which is the face that, the side of the Golgi apparatus that faces the plasma membrane. Vesicles, new transport vesicles, bud off from the trans face of the Golgi apparatus and either become lysosomes or carry their contents to the plasma membrane. Lysosomes are small vesicles found in the cytoplasm full of enzymes. There are a couple of different types of these vesicles. You have peroxisomes and then you have lysosomes. They both contain enzymes, but those enzymes do slightly different things. So peroxisomes are primarily for detoxification, neutralizing dangerous molecules, specifically neutralizing free radicals. Free radicals are molecules that really, really like to form bonds with other molecules. They'll butt in where they aren't wanted all over the place, messing up your proteins, messing up your lipids, messing up your DNA. And peroxisomes contain enzymes that can deal with these free radicals and turn them into something harmless. You have oxidases. Oxidases convert radicals to hydrogen peroxide. And then you have catalases, and catalases take that hydrogen peroxide and convert it to water. So together, those free radicals are converted to harmless water. Now, the lysosomes also contain enzymes, but those enzymes do different things. The enzymes in lysosomes are digestive enzymes, so they break down material, and this material can come from either inside or outside the cell. So they can take up stuff that has been taken up from outside the cell and break it down into its molecules. Also, if you have damaged organelles inside the cell, lysosomes can break them down and get rid of them so that the molecules that make them up can be reused. Last but not least in the cytoplasm, we have the cytoskeleton. Cytoskeleton is made up of different types of protein fibers. We have the actin microfilaments, which are the smallest size, intermediate filaments, and then the microtubules, which are bigger. And this is just a great little image that shows the different elements of the cytoskeleton. This greenish blue color is the nucleus. Yellow is a stain for the microtubules, so you can see the network that the microtubules form. And then the light purple is actin. And you can see how actin kind of spreads throughout the entire cell, particularly under the membrane, giving the cell shape. Actin microfilaments are made up of actin subunits, so actin is actually like kind of a globular-shaped protein, but it forms these spiraling filaments that are important for cell motility and for cell shape. They also form a strengthening mesh underneath the plasma membrane to help a cell keep its shape. Intermediate filaments is a category of fibers. The protein composition varies between cell types, but intermediate filaments are very strong and they resist pulling forces. So keratin that you find in your skin and in your hair and in your fingernails, that's actually a type of intermediate filaments. Microtubules, the largest component of the cytoskeleton, are made up of a protein called tubulin, arranged in a spiral. And microtubules are really the transport highway of the cell. When you have those vesicles moving between the endoplasmic reticulum and the Golgi apparatus, or from the Golgi to the plasma membrane, they're moving along microtubules. They're also really important in cell division because it's the microtubules that move your chromosomes, move your DNA from two opposite sides of the cell. This is just showing you what I mean by microtubules acting as the intracellular highway. There are motor proteins that can grab onto vesicles or chromosomes and walk along the microtubule carrying that vesicle. It's energy intensive, it does require ATP, but that's how vesicles get transported through a cell. And I want to show you that actually happening. So this is your microtubule. That's the little vesicle that was actually moving along it. This is probably another vesicle. I'm not 100% sure. All right, I know this has been a long video and we've covered a lot of ground, but you should be able to answer these questions. Describe a variety of different structures within the cell, and certainly talk about the importance of the nucleus.